So in this video, we're going to be looking now at blood vessels, the tubes that carry the blood from the heart out to the peripheral tissues and then back around to the heart again. So here we go. So the function, it's all about conducting blood out to the peripheral tissues, but the challenge is that these blood vessels have to be constructed in a way that does not let them lose any of the materials that are being carried by the blood. So, on the way out to the peripheral tissues, we don't want to be losing oxygen or losing the nutrients to tissue fluids before we're ready to do that exchange. And then, when they get to the capillary networks, it's all about facilitating the exchange of nutrients, dissolved gases, waste, so whatever, hormones, all the things that are being carried in the blood and that are in the interstitial tissues that we need to get into the blood. So we've already talked a little bit about vessel naming, but arteries are the largest of the blood vessels that take blood away from the heart. And the arterial system originates at the pulmonary trunk, so leaving the right ventricle, and at the aorta, as the blood is leaving the left ventricle. As the arteries branch, they become smaller and smaller, so the aorta leaves the heart, but almost immediately blood vessels begin to branch from it. They are also arteries, but eventually, at some point, there is enough change in the structure of the wall of the blood vessel that we stop calling it an artery, and we refer to it as an arteriole. So these are the smaller branches from the arteries, and they conduct blood from the arteries out to the capillary networks. So arterioles continue to branch and branch repeatedly, forming eventually, as they get small enough, the capillaries. These are the smallest blood vessels that we find. Their diameters may be only just a little bit larger than a red blood cell. And it's the capillaries whose construction allows the exchange of chemicals and gases between the blood and the tissue fluids. So we'll be looking at the structure of the blood vessel walls. That'll be a big piece of the work that we have to do. So finally, as the blood leaves the capillary networks, the little capillaries merge and merge until they finally form vessels that are large enough to be referred to as venules. These are the smallest blood vessels in the venous system. So when I use that term venous, I'm spelling it. Okay, so when I use that term venous, I'm spelling it like this. O-U-S. And as we talk about the arterial system, I'm spelling that word like this, not like arteriole. So these are the adjective forms of the venous system and the arterial system. So venules conduct blood away from the capillary networks, and they merge and merge, forming larger and larger venules until eventually they become large enough, and the construction of the blood vessel wall is what we would refer to as a vein. And the smallest veins are formed by the merging of the largest venules, and then these small veins will merge into larger veins and larger veins until eventually they terminate at the heart as the superior vena cava, the inferior vena cava, both of them feeding into the right atrium, and the right and left pulmonary veins that feed into the left atrium. So that's the naming of these blood vessels. And it's all based on the structure of the wall. So that's what we're going to look at next, is what is the structure of the wall. But before we do that, I want to point out a couple of small things. Blood vessels are not named based on the oxygen content of the blood that is being carried. So in the systemic circulation, and the coronary circulations, the arteries carry oxygen-rich blood, while the veins conduct blood that is poor in oxygen and relatively rich in carbon dioxide. But in the pulmonary circulation, as the blood is leaving the heart, that blood is very poor in oxygen, because remember, it is just returned from the systemic circulation and is being pumped over to the lungs to have the oxygen recharged and the carbon dioxide gotten rid of. So when the veins that return from the lungs bring blood in, that blood is going to be much richer in oxygen than the blood that was carried in the arteries. All right. Hopefully that makes some level of sense and you're with me. If that's still making confusing, please send me an email and let me know, and I can try to clarify it a little bit further. Okay, so finally now we're ready to look at the structure of the blood vessels. And blood vessels come with 3 distinct layers, and the composition of these layers is very different from each other. So the innermost layer is referred to as the Tunica Interna. So here's the Tunica Interna. And then surrounding the Tunica Interna is a relatively thick layer called the Tunica Media. So hopefully middle media makes sense to you. And then there's the most superficial layer, again a very thin layer, 1 cell thick. I'm sorry, it's just the inner layer that's 1 cell thick. The outer layer is the most superficial layer, is referred to as the Tunica Externa. So 3 layers: Tunica Interna, the innermost layer; Tunica Media, the middle layer; and the Tunica Externa, the superficial layer. And I'm trying to think. So there are alternative names for the Tunica Interna. That's sometimes called the Tunica Intima. So think it's the one that is most intimate with the blood. And then sometimes that Tunica Externa is referred to as the Tunica Adventitia. Adventitia. So those are alternative names for it. We will use, like the book does, the names Tunica Interna and Tunica Externa. I think they are most useful in being able to remember. Okay, so we'll start by looking at the Tunica Interna, and it is the smooth innermost lining of any blood vessel, is Tunica Interna, and it's very important that it be smooth. It's composed of a simple squamous epithelium. We refer to that as endothelium. It's the same kind of tissue that we find lining the chambers of the heart. So we've already talked a little bit about endothelium. And so it's a single layer of flattened cells. That's what that simple squamous epithelium means. And then underlying that layer of cells is a connective tissue basement membrane, and it's rich in elastic and collagenous fibers. So the collagenous fibers give it strength. The elastic fibers give it the ability to stretch a little bit. And that's important, especially if it's an artery that we're dealing with, because arteries do change diameter depending on the pressure that's inside those arteries. So why is it so important that it be nice and smooth, slick, innermost lining? And that's because red blood cells and platelets need to be able to slip right on through even the smallest blood vessels without sticking or being damaged in any way. What you don't want is to have some platelets come out of the blood and start collecting randomly inside of a blood vessel. That can then lead to, if a little bit of that platelet clot thing breaks off, then it could lead to a circulating embolus. So the simple squamous epithelium, that endothelial layer, is secretory and produces chemicals that inhibit platelets from aggregating. keeps them from sticking together. And it also secretes chemicals that can dilate or constrict the blood vessel. For instance, nitric oxide, that's NO gas, causes smooth muscles to relax, and that causes then dilation of the blood vessel. Okay, so there's the innermost layer, the smooth single cell layer, thick endothelial layer of the tunica interna. So as we work our way to the more superficial layers, the next layer is the tunica media. This is that middle layer, and it's composed primarily of smooth muscle, but that smooth muscle tissue is supported and strengthened by collagenous and elastic fibers, because again, the blood vessel needs to be able to expand, to absorb to stretch, to absorb the shock or the force of the blood vessel pressure that's produced by the heart. These smooth muscle fibers are arranged, so if this is a blood vessel here, and here's the opening, the fibers are arranged so that they form a ring around the blood vessel. And so that means that, if you have, now we're going to look down the barrel, there's the tunica interna, here's the tunica media, with all of the fibers arranged in this way, so that if they shorten, what's going to happen to the diameter of the blood vessel? Hopefully now you've thought to yourself, you realize, oh, it's going to get narrower, it's become smaller. The diameter of the vessel decreases, and we'll see vasoconstriction. As the muscle fibers relax, then those elastic fibers that were stretched during the contraction phase will cause the muscles, that tunica media layer, to kind of spring back to its original shape, and we see then dilation of the blood vessel. These muscle fibers are under the control of the sympathetic division of the autonomic nervous system, and when you stimulate them, then we see vasoconstriction. So stimulation causes the smooth muscle fibers to contract. Then if we inhibit that stimulation, we stop the stimulation, then we see vasodilation as all of the muscle fibers relax. This is there's a drawing in the book, and I don't have my book with me, so I can't tell you what page number. But there's a drawing, and then there's a picture of a blood vessel under a light microscope, and it shows very clearly how much thicker the tunica media is in an artery than it is in a vein. And that's important because it allows the arteries to resist the blood pressure of the blood that's being pumped out of the heart. So the tunica media is primarily smooth muscle, a little bit of connective tissue in there as well, the thickest layer of the blood vessel. So now we're going to go to the outermost layer, which is the tunica externa, also known, as I mentioned already, as the tunica adventitia. It's a relatively thin sheath of connective tissue that's composed of elastic and collagenous fibers, and they surround the blood vessel, and they give it a little bit of extra strength because of those collagenous fibers, and again, some more elasticity because of the elastic fibers. And the role of the tunica externa there is to attach the blood vessel to whatever surrounding tissues there are, to hold it in place in those tissues. So there you have it, the blood vessels of the body and their names, from the great arteries of the heart that branch and branch to form smaller arterioles, and then the very smallest of the blood vessels, the capillaries, where we finally can see some gas exchange. We're going to look a little bit more at that in one of the next lectures. And then on the far side of those capillary networks, the capillaries merge and merge to form venules. The venules then merge and merge to form the great veins of the body. And each of these blood vessels is composed of 3 layers: the tunica interna, which is a single cell layer thick. It's built of endothelium, And then the tunica media, which is the muscular layer that can either constrict or relax the blood vessel, causing either vasoconstriction or vasodilation. If you're hearing noise in the background, that's our dishwasher. It's draining out. Sorry about that. And then finally, the outermost layer, the tunica externa, which is connective tissue that forms a sheath around the blood vessel and holds it in place in the tissues. Okay, so there we've got it. And we'll move on to the, excuse me, to the next